Hello and welcome to Science on Trial and Error. In case you haven't been here before, I'm Kasia and I'm a PhD student in developmental biology at IST Austria. In this podcast, I chat with researchers about their lives inside and outside of the academia. Why? Because I believe it's important to share our stories, especially now when the usual ways of meeting new people are very limited. And who knows? Maybe my guest stories will inspire you to try something new or help you with finding your way in science. Or maybe it will bring you comfort of knowing that a lot of the other scientists out there share your everyday struggles and difficulties. Whatever the reason, I'm glad you're here, and I hope you will stay for longer. Okay, this is very exciting. It's already episode number 3, and today my guest is Maciej Kerlin. He is a Polish researcher currently doing his PhD at the Institut Curie in Paris, and he's working in the group of Antoine Coulon on enhancer-mediated gene co-regulation within the 3D genome. Me and Maciek have actually met during our bachelor studies in biotechnology at the University of Warsaw, and even though we officially continued our studies there for the masters, we were doing our research externally and didn't overlap so much. So it was great to catch up with Maciek. Besides his lab work, he plays violin and loves traveling. As a PhD student representative, he's very committed to improving the lives of other students, and somehow he still finds the time to take on new initiatives. But find out more for yourselves. Enjoy, stay in touch to let me know what you think, and please welcome Maciej Kerlin. Hi, Maciek. Thank you for accepting the invite to talk on my podcast. It's nice to see you after a while.
Yeah. Hi, Kasia. Nice to see you too. And thank you very much for inviting me. And especially at so early stage of developing your podcast. So it's a pleasure to be here.
Yeah, I'm very excited to talk about your work. And I'm very excited to hear what you've been up to since we've been studying together. So it's been a while. So you're currently in Paris, right?
Yes.
And how is the situation there with the COVID?
Since today we've entered the third lockdown. Basically, there were 2 before, and they were like nationwide. So the whole France was confined, and this time they went more towards the approach with like closing just the region. So basically, the whole region of Paris and Ile-de-France is closed right now. But to me, it didn't change much, basically, because I can still go to work. I can still go out, do shopping and all stuff like that. So fortunately for me, what was actually the most important for me was that they didn't close our institute. So I didn't have to stay at home again. Because the first lockdown, it was like 2.5 months that I stayed here. And we were not able to go to the lab. So that was quite challenging time. And I would prefer to not do it again. So I'm quite happy that I'm still able to do my work. So
yeah, I can understand that. So you're currently writing your thesis, right?
Yes, I started some time ago.
And how's the writing going?
It's slow. It's very slow, especially that I'm still doing my experiments. So trying to balance the time between doing the experiments and finding the time to write, it's challenging, basically, but I think I'm getting better at that. So I try to have at least one day per week where I don't do any experiments, no analysis, nothing like that. I just stay home and try to focus on writing. And I think that's the approach that I'm going to take.
So what I like to do at the beginning, just to kind of relax, is to talk a bit about what helps you focus to work in a lab. And I was wondering if you have any kind of music that you like to put in your headphones when you're working, or maybe you're listening to like podcasts, or what is it that keeps you focused or actually keeps you in a mood to do the work?
It's totally music because I listen to podcasts as well, but I feel like hearing other people's talk, it distracts me. So I totally prefer to do the music. And for me, it's actually the classical music. I know a lot of people actually do classical music to help them focus. And me recently, I found that very nice violin sonatas of Schumann, and I'm a huge fan of those.
You're actually playing violin, right?
I do, yes, yeah. I mean, now, since I moved in Paris, it's a bit less. And also because in the building that I live in, the walls, they are kind of made of paper. So basically you can hear everything what my neighbors do. So I also try not to be too annoying for them, especially that we are on confined and people spend a lot of time at home. So I just try to be respectful as well. But yes, I have my violin here, and whenever I can, I try to play. Also, it helps a lot with relaxing. So
yeah, I think classical music has, it also doesn't have the text, so it doesn't distract you with the words.
Exactly. Yeah.
But it can really change your mood quite easily.
Yeah,
and
yeah.
So you are working in Institut Curie, right?
Yes. Yeah.
And it's the group of Antoine Coulon.
Exactly.
So how about we talk a bit about your current project? Can you tell me more? Because I'm actually very curious. I just saw the title, so let's go into that.
The project that I'm doing right now is basically focused on the three-dimensional genome organization. The average length of DNA coming from just one human cell is about 2 meters. And this DNA has to be packaged into the very, very small volume of the cell nucleus, which is about a few 5, maybe to 10 microns in size. So how do you fit this 2 meter long strand of DNA into such a small volume? And at the same time, all the processes happening in the nucleus must happen as well. There's DNA replication, DNA repair, gene expression, basically, and the timing of gene expression, like expressing right gene at the right time. So this has to be very precisely regulated. And so this kind of imposes another constraint on taking into account this whole length of DNA that must be packaged into the nucleus. And what I'm investigating is basically how this packaging of DNA in the nucleus, how does it affect gene expression? I'm mostly interested in regulation of genes by enhancers. And one feature of this three-dimensional organization is that there is a specific level of this organization which spans the range of kilobases to megabases. And within this range, there are three-dimensional structures which are called topologically associating domains, or TAD. So basically, you can imagine the TADs being, if you think about this 2 meter thread of DNA, you can think of TADs as a small knots at the length of this whole DNA. So basically, what I'm looking at is how enhancers communicate with genes inside of those small knots, and how gene expression is actually co-regulated within those knots. So basically, what we are using in our lab are the imaging approaches. And what I do is the fixed imaging approaches. Okay. So all kinds of fish, both DNA and RNA fish.
But still, the scale that you need to look at is very small, right? So
yes.
Do you need to use a special microscope, or is it possible with like normal confocals?
No, so we don't use confocals. We just use the regular wide field microscope, because for the fish approaches is quite good. And I'm not yet at the scale where you actually need the super resolution microscope, because those are the very special ones that let you go below the diffraction limit. But actually for what I need, just a regular wide field microscope is okay.
Okay. So you are doing the fish, which is the fluorescence in situ hybridization, just for the people who are not biologists.
Yes.
And you are looking at the genes that are co-regulated by the same enhancers within this TAD regions, right? This is what you said.
Yes. Yeah.
Okay.
Yes,
so
exactly.
This could be actually, because it's a knot, the genes that are not super close to each other in the DNA, but because of this formation of the knot, they get closer together, right? Am I understanding it correctly?
Yes, I mean, like if you think about relative genomic distances between those genes, they are also quite close to each other, because the size of those stats, it goes up to 1 megabase.
Okay.
So 1 megabase actually compared to like the whole chromosomes that can be like hundreds of megabases is actually relatively small. So still, it's true what you said, that they come close together in 3D space, but it's also true that they are relatively close to each other in the genome.
So are there any specific genes that you are looking into? Or is it more general question?
So I'm working on one very specific locus because the constraints that we put on ourselves before starting this project were there was a whole list of criteria basically, but one of the criterion was genes to be regulated by estrogen. Mm-hmm. So the model system that we use is the breast cancer cell line MCF7, in which you can just add, the estrogen, the hormone to the media, and then many genes of those cells will become upregulated. So I studied the locus with 3 genes that are upregulated by estrogen, and they actually reside within 2 adjacent TADs. So I have 2 genes that are in one TAD, and 1 gene that belongs to a TAD that is just neighboring to the first one.
And they are all co-regulated because they respond to the same inducer.
So that's actually a very interesting question, because what we are trying to understand is the multiple levels of co-regulation. If the co-regulatory mechanism, if you think about it as a hormone, as estrogen, then you can say that, the thousands of genes that get upregulated or downregulated are co-regulated because they all respond to estrogen. But there are also more local levels of co-regulation that are provided by TADs, by those 3D structures, because from the literature, we know basically that, for example, during differentiation of mouse embryonic stem cells, so that's something that other groups have shown, that during differentiations, genes from the same TAD show this co-regulation. It's a feature that has been observed for TADs as well. While we try to understand both of the stimuli, on one side this induction with estrogen, and on the other side this local effect of that makes it a bit more complicated, and that's why we try to understand how all those different things might affect the co-regulation that we see. And that's probably not all the factors that affect the co-regulation, because we can try to understand how estrogen affects this, how TADs affect this, and then within TADs how enhancers affect this, because enhancers can skip some genes. You can have, let's say 5 genes in a TAD, and one enhancer might regulate only 3 of them. So then what happens to the other 2? There are really many levels that we try to disentangle, and it's not always the easy task.
So the formation of these TADs, how is this regulated actually? Is it something that is epigenetically kind of regulated based on the structure of the chromatin, or is it sequence based? How does it work?
Since few years, basically the most popular explanation for formation of TADs is a process called loop extrusion. So basically at the boundaries of those TADs, there are specific sequences which can be bound by an architectural protein called CTCF. So basically if you have a chromatin fiber, the ring-shaped cohesin sits down on it and start extruding the loop, basically, until it reaches those convergently oriented CTCF sites bound by CTCF. And binding of CTCF creates kind of a roadblock on which the cohesin ring will stop, and therefore the TAD or loop will be formed.
So this is very recent research.
Yes. So basically, this the whole field in general is quite young.
It's very hot,
because it's. Yeah, exactly. A lot of things are happening, and basically the first techniques that were developed to study these kind of questions, it's the matter of the last 2 decades. So it's really a very young and dynamic field. And when talking about loop extrusion, we are really talking just about the last decade. So that's a really very recent.
This is amazing.
Yeah.
This is amazing.
Yeah.
So, your research currently is PhD project is kind of ending because you have to write your thesis. And
yes.
How did it work? You applied to Curie, and your studies had a limited time of contract, or?
I basically applied to the Institut Curie International PhD program, IC3I. And while I was applying, basically there was a list of projects that I could choose from, and I just decided to apply for the one I'm doing right now. And the funny story actually about applying to this project was that I was looking for a PhD position and I was checking like many institutes, many different programs. And actually I came across of the Curie program twice. And at the first time, like I saw the titles of those projects, the very first selection was I look at the title and does it seem interesting or not? The title of the original project was like crazy long. It was like 3 lines of text. And I basically rejected that after just seeing this 3 lines of text because I could understand nothing out of that. So after the first attempt, I didn't even think anymore about applying to Curie. And then maybe like 2 months later, actually it was just a few days before the end of application process, I came across again. And this time I didn't only stop at the titles, I went more into the description and I found, actually, it's not that bad. This project seems very nice. And a link to epigenetics and the genome studies, which was something that I was interested. So, yeah. So I applied, and after the first round of selection, I got a reply saying, okay, that your documents are fine, so you can go into the second round. Which were the interviews. So I got invited to come to Paris for the almost a week interview session, which was very nice. I was really impressed by the whole organization of that time. Yes, so a few days after those interviews, when I came back actually to the US, because at that time I was living in the US. So a few days later, I got an email saying that, okay, you are accepted. So welcome to Paris, basically.
That's amazing. I mean, I find these interviews really, really good thing because you can just see the place and just get a feeling. Like,
yes,
of course,
exactly.
the interviews itself are important so that you can meet the PI. But I think the whole process of going to the place helps you make an informed decision, which is why I feel bad for people that have to apply now because of COVID. It's much harder to get the feeling of the place,
Exactly, yeah.
and to see how this works. Before we discuss more about Paris, I actually want to go back a bit in time. So what I also like to ask people is, how did your love for science and for biology started? Because I know that you have been actually quite focused on the epigenetics and on like the genome regulation throughout your whole, let's call it like bio career.
Yes.
But what actually drove you to this point? Did you have a very influential teacher, or was it something that was actually in your house? Like, was science important in your house?
So actually, I was just recently thinking about that. And I think like my interest in biology in general, that started already in the primary school. But I remember that while being a very small, like maybe 7-year-old, so I didn't yet have any like particular knowledge about biology or anything like that. I just got this plastic microscope. It was a very, very simple microscope. It was basically a toy, but it had this small mirror that I had to adjust properly to just be able to see anything. And I remember my mom, who was at that time basically the director of the school, she contacted me with the biology teacher from that school who gave me a few samples. So I was able to observe. And that was actually, I think, my very first experience with science. But I must say that, during primary school, it was not this love for science. It was not there yet. Basically, I think it started really on the later stage. At the same time, I was also really interested in like tourism and geography, and I was always telling myself that I'll become a geographer. Or And actually, when I went to the high school afterwards, I went to the class that was profiled for on geography and economy. So that was not really something related to biology at all. But somehow this interest in biology was still there. It was not just kind of my primary interest at that time. And I was very lucky to meet another amazing biology teacher in my high school from which really the serious thinking about biology started. And I remember very precisely when was that, because it was a class about biotechnology. And what biotechnology does and how is it helpful in today's world. And I remember it was the first time we were learning about a PCR technique. And I was so amazed, how is it possible to just, amplify the DNA and, you just put a little bit of DNA and then there's this 1 or 2 hour procedure after which you get a tons of the same identical copies of DNA. And that's what I found was amazing. And this is really the moment on which I decided that I want to study biotechnology.
Actually, you come from north of Poland, right?
Yes, yeah.
And then you moved to Warsaw to study biotechnology at University of Warsaw, where we actually met.
Yes, exactly.
And and there you were working in the group of Professor Jerzmanowski, right? Mm-hmm. So you were working on chromatin dynamics in plants, right?
Exactly this, yes, yeah.
So how did you like the biotechnology studies in the end? Was it what you were hoping for?
Yeah, exactly. So in the end, I liked a lot what I got into, and the project that I did in the lab of Professor Jerzmanowski about plant chromatin and epigenetics. But I think when you really look at the very strict definition of biotechnology, it's not what was that interesting, because as you remember as well, we had courses on biotechnology and how to apply biological processes in industry. But that in the end was not something that was like really interesting for me. I really started to like this, basically what happens in the nucleus, so all this genetics and epigenetics. So that's what I found most interesting.
So why did you decide to actually, were you interested more in the genomics and genetics, or was also something there that was driving you towards plants, or plants was just
No, that was actually not. Plants was not the very first choice, because I remember there was this idea that, who studies plants? It's so boring, like nothing interesting happens in there. But to me, I think it was like the thing that I liked about epigenetics in general was that this whole array of techniques that you can use, like starting really from genomics approaches like ChIP-seq, RNA-seq, all the way to the imaging. So I must say that since I had this very first microscope, I always like to observe things. Yeah. And that's why basically when choosing PhD, I try to go into imaging labs, because I just like to see things. And I think doing microscopy is just the easiest thing to see things and not try to, infer based on some other result. Mm-hmm. Yeah, coming back to this genomics, epigenetics, that was, I think, again, that this variety of techniques was something that was interesting. And since it was in plants, then I just decided, whatever. I can just go and learn all those techniques, and then I can apply them like anywhere. And I don't think that plants are boring at all. So
actually, from the genetics point of view, plants' genomes are very interesting, right?
Yeah, exactly. Because plant genomes, like compared, for example, to the human genome, like plant genomes are much, much larger.
Exactly.
Yeah, that's totally a very interesting model to study.
Yeah, I agree. So you did your bachelor's there, and I think I remember that during your master's, you actually went abroad for a while, right?
Yes, I did. So I applied for an Erasmus exchange, mm-hmm. which happened actually also to be in Paris. And there is also a funny story about that, because I remember there was a list of the universities that our faculty had had agreements with. So it was not just in France, but I remember Spain, Sweden, Germany, so there was plenty of choice. But I always wanted to try to live in France, because I've been learning French for quite some time. But the university of my first choice for Erasmus was not at all in Paris. It was somewhere in the Lorraine region. It was basically dictated by the high cost of living in Paris, because I read before, like while making my choice, I was taking into account like how much I can afford with the stipend that I get from Erasmus. So Paris was not at all my first choice. There was an interview and I got accepted, and I picked the Université de Lorraine as my first choice. And I remember on the day that the Erasmus coordinator at our faculty, when she had to send all the papers, she wrote me an email saying that, Université de Lorraine, they withdrawn from the agreement with us.
No.
So you have to choose something else. So I just told her, okay, I'm taking Paris and just still send my application. And so that's how I ended up in Paris. And a nice thing about the project that I was doing was that Professor Jermanowski, my advisor during bachelor's and master's, he knew another PI here in Paris at the Université Paris Sud and INRA, which is the French Agricultural Research Institute. So they decided to conceive a collaborative project just, to be able to study things that she was interested in her lab and things that we are interested in our lab in Poland.
So this ended up being your master's thesis?
Exactly, yes, yeah.
So for how long were you in Paris, actually?
It was 6 months. It was
that's quite short.
Half a year of exchange. Yeah, it was quite short, but again, thanks to the fact that we were talking about this project, because, the recruitment for Erasmus, it was happening already quite some time before I actually went in Paris. So we had the time to discuss the project, and because, with plants, the thing is that they need time to grow, they need time to produce the seeds. So I actually spent that time before actually going to Paris to produce some material that I could later use. And since my exchange was at the first year of master's, then I still had the whole second year where I could continue basically this research to be able to make it my master's project, which became quite big in the end.
So this was also chromatin dynamics related research, right?
Yes, so this project was really going into mechanisms of how chromatin is dynamic and how this dynamics is mediated by linker histones and HP1 protein homologue in plants, which is called LHP1, which is basically involved in the formation of inactive chromatin, the heterochromatin.
So were you doing also a lot of imaging during this project, or?
Yes. So a huge part of that project was to image the nuclear distributions, relative nuclear distributions of this LHP1 protein and of H1 histone variant.
You went to Paris it was a bit lucky, but in the end you still ended up kind of going consistently in this direction of your interest in genomics and genome regulation. And then you finished your master's, but you actually didn't apply right away for the PhD, right? You went for a year to US, which is also super exciting.
Yes.
So you applied for this Fulbright stipend? Was that?
So yes. So during the second year of my master's, a lot of people already from our faculty were deciding, okay, are we staying in science or not? And some knew right away that they will leave science and go to work into industry, whereas other ones were already applying for the PhD. And I still didn't feel yet like, do I already want to go to PhD, or I just prefer to explore something else? And at that time, I was not sure about that. And there came this possibility to apply for this Fulbright program. It had a very, very long name. It was like visiting research graduate traineeship program, something like that. So I just applied. I just thought I would give it a go, and this would give me some more time since this program was running for a year. So I thought this would give me some time to decide what I want to do after the university. In the end, I ended up being invited to Oklahoma Research Foundation in Oklahoma City. The project was actually not, it was still in the nucleus. I still got into the lab that was studying the DNA replication in zebrafish this time. So not only I changed the subject a little bit, but also the model system.
First of all, from what I'm hearing by now, it seems like your path was very, very consistent. Like you really stayed within your topic of fascination, which is more related to genomics and nucleus and the processes. And on the other hand, you were kind of trying to gain more experience with trying different techniques and trying different maybe systems a bit to really see what interests you, right? So you said the yearly traineeship or whatever internship that you did in US was supposed to give you an idea whether or not you want to stay in science. And, afterwards you apply for your PhD. So I guess it really persuaded you. Was there something in particular that really pushed yourself towards the PhD? Or Was it just the research that was new, or what was the reason?
I think it was the possibility of, still being curious and still trying to explore new things, because at that time, because this was really like quite a long project and this was really like, because we were working on the paper at that time as well. And I think it showed me how, if you have at the beginning of each project, you have a very specific goal that you are trying to reach. And then during the project, there are like unlimited number of like side projects that open. And I think that's what got me to stay in academia and to pursue the PhD, like this fact that there is so many possibilities that open while during your project, that basically the possibilities to explore are endless. There is going to be always something new, something unexplored that you can try to look at.
Yeah, I think it's important to realize this, and I think sometimes during your studies you are so a bit stuck in what you're doing that you don't realize how much is happening. I think conferences help in that, but of course getting a different research experience also can open your eyes a bit. But touristically, you also managed to travel quite a bit.
Yes. It was thanks to the contract that was constructed in a way that, and also the goodwill of the PI that I was working with, because I can imagine that they take a new person and, might be hard for them to let this person to go and not focus on work that much. But during my path, I was always very lucky to meet very nice people that value actually the work-life balance, and that allowed me to also pursue my passion for traveling. So
that's super important. So what was the most memorable trip in US?
In the US? Oh, that's hard to say. But I think I would say the trip that we did, it was actually almost towards the end of my stay in the US, where we went to California. All those beautiful natural parks that are there. I think I was most impressed by Yosemite, because we did a day hike in there, and it's really, really a beautiful place. So yeah, I think if I were to choose which one was the best trip, I would say it was that one, probably.
That sounds really cool. So it seems like you really like the work there. Were you actually considering trying to apply for PhD in US, or was it something that you didn't really want to do?
I thought about this for some time, but when I realized that, the academic system in Europe and in the US or Anglo-Saxon countries in general are very, very different. In the US, the thing is that when you finish your bachelor, you can go directly in PhD, which on the other hand takes very long. It's not like 3-4 years as it is in Europe. It's really I think 5 years minimum. And the first 1 or 2 years you basically spend taking classes. So it's kind of the beginning of your PhD in there is what we call as a master's here in Europe. But I kind of didn't want to get myself into kind of doing masters again, since I had it done already, and I thought it would be a bit of waste of time. So that was one reason. And the second reason was that most US universities, they require you to pass first the language test and GRE. And the thing is that it's crazy expensive. So I was not willing really to pay for that. And second thing is that GRE is really a very particular test in which they do not only, it's like a general knowledge test basically, and there is a language part in that as well, in which you have to learn on average 3,000 of words that no regular English speaking person even uses. So I found it was a bit too much of an effort for staying there for 5 years at least. That was also the one thing that I didn't want to stay away from my family for that long.
Sure.
And going through all these tests, paying all this money just to spend first 2 years on repeating what I did in my master's. So in the end, I thought that staying in Europe would be a better choice than that.
So you were looking for, as you said, you had a specific scientific interest. You wanted to work on chromatin dynamics. If I remember correctly, because I actually also looked into Curie when I was applying, is that you apply for a specific project that actually already has been kind of pre-designed by the PIs. So was it something that you actually liked that the project questions were kind of set already and you could dive into work, or was it something that you didn't really care about when you were looking?
No, so that's actually not something that I cared much when I was looking. But if there were programs that would require me to really design a project from the beginning, I don't think I was at the point, and I had like enough of knowledge to be able to design the project from the beginning myself. So actually the fact that it was already pre-designed, it was very nice. And also thanks to this, I had a description and then I could see, okay, so that's really something that I could do, right? That was a very good advantage, I think, at that particular point.
So the project is designed for how many years? 3, 4?
Yes, so you get in the IC3I program, you get funding for 3 years, basically, because that's how much money the European Union gives. And the rule in France is the basic timing for your PhD is also 3 years, which is on the lower side.
That's
actually,
quite
when you compare
short. That's quite short.
With many European countries. But you can get extensions for 1 year. But that's it.
So I know that a lot of these programs that are international programs, especially if they are set actually in French-speaking countries, even though the basic language is English, there's a lot of things that happen in French. So was it actually something that you were finding difficult, or because of your knowledge it was kind of fine to adjust to working in a French-speaking institute? I mean, I know that Curie is international, but still France tends to have their own rules about that.
Yeah. I mean, that really depends on the lab that you end up being with. So, because, Curie is composed of, there are basically 3 sites. The one that I'm working in is really in the center of Paris, but there are also 2 other sites, mainly hospitals that are located more in the suburbs, but there are also researchers working in there. But I would say it really depends on the lab that you end up being with, because in my unit, the one that I work on, it's crazy international. In my lab, basically the only French people is the PI and one of the engineers that we have, and we are 8 in the lab in total. So my lab is totally international. But I know also the labs that have the French only people, and there I can imagine that that it might be a bit difficult to adjust. But I think also on the other hand, PIs do a lot really to be welcome and to make people feel welcome and not feel alienated because of not knowing the language. Because for me, as you say, it was not much of a problem because I could speak French before.
Yeah.
But I know people just coming here, it might be very overwhelming to try to adjust. It's not maybe that much of a problem inside the institute, but for the everyday life, the French are really proud of their language and they use it a lot.
So, okay, I think research-wise we discussed a lot, but I would like to talk a bit more about what you do outside of research. And I think it will be still research kind of touching, but first of all, what I find very cool is that you try to be very involved. From what I saw in your resume is that you are not only a PhD student representative, but you also established this network for imaging researchers in France, right? So maybe you can tell a bit about this and we can make it a bit more known through the podcast.
Yeah, so there are many extracurricular activities that you can do. So as you mentioned, I'm basically every unit has their own lab council. So this meeting happens several times a year, and then this is really the time to discuss what are the current things happening in the unit, typically audio renovations, for example. That's always a huge discussion in our unit because we are working in basically the historical buildings where Marie Skłodowska-Curie used to work and do her research. And me as a PhD student representative, I'm really focused to try to bring up the problems that we are facing as PhD students, and especially now during pandemic, this
it's
we
special
are basically
circumstances.
the most affected and the most vulnerable group in the whole, hierarchy of the academic research. Yeah, so that's one thing that I've been involved in. And the second one that you mentioned is this network of young imaging researchers in France. Every 2 years, we have this meeting, which is called MIFOBio, which stands for Functional Microscopy School. So, and we basically spend there a week. And this is week of full intense lectures, courses, practical works on the microscope. It's really a thing where people from whole France come in. There is a special room dedicated for doing the cell culture. So basically, there are hotel rooms that are made the cell culture with the proper hoods, proper incubators, and everything. There are people who bring €1 million worth of microscopes with them, and you can bring your samples and you can test them. If, for example, you want to learn
so cool.
super resolution microscopy, you can test the newest and craziest microscope that's on the market, for example, and you can see if it helps to answer your question or not. And yeah, we thought that it would be nice to have some space also for young researchers. So we just decided to organize conferences dedicated strictly to this topic of imaging in biology. Not only in biology, because we also have people who are interested in biophysics and chemistry, optical development, and microscopes development. So there's this whole diversity of topics for which imaging is useful. And I think it's nice that there is this young researcher community going on. So far, we've organized 2 conferences already. So one, which happened in Curie 2 years ago, which we could still organize in person. And last year, since it was already during the pandemic, it happened in Institut Pasteur, also in Paris. And this one we did remotely already. But still, I think it was a very nice and very actually interesting and teaching experience to try to organize such a meeting in pandemic and completely remotely. So yeah,
it's not easy. It's not easy to do this. What's the name of it's called IMA Bio Young Scientist Network, right? Just in case someone wants to check it out.
Yes.
This is great. I mean, I think it's really important to get involved in such initiatives, and yeah, it's very good also for you, right? Because you get to know more people, you network, you know what is cool, what other people are doing, and you just stay on top of what is cool in the field.
Exactly, and this also fosters a lot of collaboration. There are many collaborations actually that happened through these meetings. It's also very good, for example, for looking for a postdoc. Then if you have a position, for example, you can just advertise it within the whole network, and then almost every time there is someone from the network who is interested in that. So yeah.
So speaking of postdoc, are you looking? Have you decided that you're staying in academia?
Yes, so I will stay in academia, at least for a postdoc time. I know that there are a lot of people who, even after a PhD, who want to leave, and some of my colleagues also are considering to go into industry, but that's not the case for me, so I'm planning to stay, and I'm looking for a postdoc. Not that much yet, because I have this comfort that after defending my PhD, I will still be on the contract here in my lab, so for a few months. So
to finish the paper.
Yeah, exactly. So I'm planning to dedicate that time more to look for some possibilities. I mean, I have some labs already in my mind, and yeah, we'll see what's gonna happen.
Are you planning to continue in the same direction? So like chromatin.
I think so. Yes. I know a lot of people think that it's good to change the topic a little bit for postdoc, but I think that for me, since. So the first thing is that when I came into this field of the 3D genome, it took me quite some time, to be honest, to get a grip on what's going on in the field and get familiar. And I'm not sure I would like to do this again while switching to a postdoc. So
I think seeing as it is very, very new, as you said, it's a very hot topic. There's constantly a lot of new things coming, so it can still feel like you are in a very new environment. Even changing the lab and getting to learn the techniques, and with the field developing so quickly, I think you are still kind of you will have still a lot of development and a lot of like self
Exactly.
learning involved. What are you also looking for? Are you looking for something that will have the imaging as well? Are you still kind of very focused on?
So that would be nice. But then if it would be imaging, I think I would try to move to live cell imaging.
Mm-hmm.
And if not, I would probably go to some genomics lab that does high-C. So high-C is one of those standard methods to investigate the 3D genome. And I think having a combination of skills, on one side doing the imaging of the 3D genome and on the other side having this knowing how to do those genomic approaches, I think that would be also very nice.
Yeah, this sounds really cool. I mean, I'm sure there's a lot of group X3 that now are like going into this direction, so there's a lot of options for you.
Yeah, especially that even at the conferences, that's what I find interesting is that there are like people from high-C labs that present, and then someone tells them like, oh yeah, you could like try to investigate that by fish. And then they say, oh no, we are not really the fish lab and so on. So that really...
There's no overlap.
There's not much overlap, and there's not many labs that do both actually. So.
But this is a cool niche, even considering the future.
Exactly, yeah.
Like if you decide to stay in academia even longer, this is something that you could proceed.
Exactly. And that's where I think the field will be going now. On one side, it's not only like that much focused on biology, but there are in the field, there are also a lot of physicists and computer scientists that my lab basically is, that I'm working at now is very interdisciplinary. Because in the lab, there is me and there is one other biologist, but we also have like imaging engineer, and we have biophysics part, and we have the polymer modeling part. So we really have like very diverse array of people that work on the similar questions from different angles.
Yeah, I think this is also the future, like you need a different perspective to be able to maybe properly approach the question, or maybe just get the new angle at the question that actually can provide some new information. So I think this is really cool. Okay, so I think there are like two things that are left that I usually like to ask. One is a bit, let's call it a serious question, which is related to the fact that, seeing as we actually go through all these stages of academia and you want to stay in academia for your future, I was wondering whether we could maybe go into what do you think is something that is still not working really well? And something that you would like to see improved. Either you want to work on it yourself, or you just feel like it's something that is still lacking and it would be good to kind of bring the attention to it.
Yeah, so I think there's plenty of things that can be fixed in academia, but I mean, especially for young researchers, I think funding is like the most basic and the most important thing is the availability of funds, and also all the administrative burden which is related to obtaining those things, because I see like plenty, including my PI, plenty of young PIs that spend their time, a lot of time, like trying to get grants, and I think this. Not only trying to get grants, but all the administration, all the reports that they need to fill, and motivating, wanting to buy a computer for the lab and needed to motivate it because they cannot, with some particular grants, you cannot buy the equipment because then it won't be used only for that particular grant. I think this is something that really needs to change, because it takes crazy amount of time that you could really devote to improving science and going further, instead of spending it on writing motivation reports and so on. So
yeah, I think this is very like sometimes it's kind of missed by the institutions, but really the amount of time that the PIs spend on this, it's so much of their time. Like they really lose track of the science, and they have to spend like half of their time just dealing with the bureaucracy. I think there are some institutes that are going in the direction that you are mentioning. Like I've heard, for example, the Janelia Institute in the US, they have their own funding that is kind of fixed for a certain amount of years, and their PIs don't have to apply for any kind of funding. They can just really focus on science. And, looking at their track record, they're doing brilliant research. And really,
exactly.
when they are able to focus on the research, it can actually move things much faster.
And they are very, very well known in the imaging community, like Janelia Dice. So it's, yes.
Exactly. They are doing, very cutting edge things, and they are really fostering collaborations strongly. But I think the fact that their people don't have to worry about funding is just, it puts you in a different state of mind, and you can, I think, especially when you're doing basic science, because, we always have an aspect of applicability to our work. Somewhere, like in your work, it can be related to cancer, it can be related to development. But in the end, on a day-to-day basis, we are doing basic science. And these are basic science questions. I think that's
really interesting what you're saying about this, balance with applicability, because I have a bit of feeling that a lot of actually funding bodies, that they require you to prove, the applicability of your project. And that's something that I hate, to be honest, because, science started because people were curious and wanted to just know the nature of things. And not their primary motivation was not necessarily, to cure diseases, which is still very important. And I can totally agree that a huge amount of money should go for that. But I don't think that this should be connected with like giving less and less money every time for doing just the fundamental research.
Yeah, I think this is the problem, that, you always have to prove how, applicable your research is. And then, of course, we always can apply it to somewhere. But the basic research is what pushes a lot of these disciplines forward. And we need it. And it is... it is harder to get money when you're doing basic research, and it is, sometimes it just comes down to feeling like you really have to push to find some sort of connection to something to get the money. And it's I don't really think this is how it should work. It's hard to fix it. But I think the more people speak about it, and the more institutes try to become different in their way of funding, they are trying to get some sort of funds kind of fixed so that they can sustain their researchers, it's going to move in the good direction, hopefully. I mean, we never know.
There is also a role for us, for the scientists, in that, because I can totally imagine, because, it's the politicians basically who decide how much money will go for basic or applicable research. And I can understand that for them, it's easier to give money, to someone who will say, oh, I will cure cancer, or, oh, I will try to work on this disease, versus someone like me who will say, okay, I want to investigate correlation of genes inside TATs and inside the 3D genome. So, but there, I think that there is a huge niche for us, for scientists, to just speak about this in a way that people can understand. Because without that, this attitude of politicians and of society, I think it will not change.
Yeah, I think this is something that I think all of my guests come down to in the end. The scientific communication is a crucial thing. And to be able to describe your research in a way that the person who is not a scientist can understand is super important.
Exactly.
And this is how we can change the way that society views the basic science, that it can still change lives. It may be not as direct, but it does move the science forward. And it's something that I think a lot of people tend to push aside. And, in the current situation with the media being so loud-spoken, but not always so well informed, it's even more important. We have this responsibility to try to at least educate, or even just explain how things work from our point of view. Yeah, I agree with this completely. So do you find it difficult to explain your work to your family and friends that are not scientists?
Yes, a bit, yes, because the first question is like, okay, so are you gonna cure cancer with that? So that it always comes down to this question when I'm at home and where people ask me about that. So then, when you're gonna cure cancer or that? Because I also say that okay, my model system is breast cancer cell lines. Many people think okay that I'm gonna just cure breast cancer with what I'm doing. So that's really hard to explain like how things work and why is it also important, to explore those many ways and just to know the nature of how things work to be able actually to push forward the application research.
Yeah, I agree. Okay, so I think we are closing. It's just one more question that I found a funny question. Let's consider this scenario that you can have a dinner with anyone who's either still alive or actually is already dead. And you can just ask them your burning questions, or you can talk to someone who really inspired you. Who would you choose?
So many people, and whether I would pick someone who's living or not.
We can go with
I think I'll just go with a cliche that I think it would be Marie Skłodowska-Curie in the end, that I would ask like how, because she was a completely pioneer woman, like she did so many things that no one at that time would think that a person would do, or even more that a woman would do. Like she was the first one to do so many things, to be the professor at Sorbonne, to get Nobel prizes and all like that. And I think I would ask her like how she thinks that the thing would go, and if she like was brought back to live to be alive today. I think I would ask her like what does she think, how the things went since the time that she was living and she was doing her research. So I think it would be that, probably.
I think this would be really cool conversation to have. I mean, seeing exactly as you said, she was a pioneer. I like to also think of her, in the sense that she was a female scientist who got extremely successful, but she also had a family, and this family turned out to be extremely successful as well. Yes. So, she was doing it all and really she gained recognition, which is really important. But I would be interested in hearing their side of the story as well. Yeah. Okay, so I think with that, we can finish. I really wish you all the best with the thesis writing and with the defense that is coming in September, you said, right?
Yes.
And then looking for a postdoc, I hope you get really interesting offers and you find a place that will allow you to grow and to still look into science and to maintain your curiosity. And thank you so much. It was really cool.
Thank you again for invitation. Yeah, that was super fun. And good luck for you as well, because for you also is it coming? Not that quickly probably as for me, but...
Yeah, it's coming, but I still have a bit of time. So.
But also a lot of luck for the podcast. I hope it will keep on, and that we'll hear more interesting guests soon. So...
Thank you. And I hope you will follow the other episodes and you will like it.
Yeah, for sure, yes.
Thanks.